basically what you need to study today, we will be working on, is on morphogenetic movements. So it's like movement of cells. The word morphogenetic means there is some movement of cells and which causes a change in morphology, appearance changes. So this is a note. This note which I have sent. This is a very brief note, but I'll be giving you a detailed note. It will come later. But before you go with a detailed note, you have to read with smaller things. Now let us start with uh, the development of and amphibian frog embryology. These are the events till the formation of the gastrula. Those who are online, can you see this?
Yes,
Yes, sir.
sir.
Okay. So this is for background studies. You need this. As you know that the eggs are normally large. The frog egg is very huge. In the early mornings, if you go out during the rainy seasons, see small puddles or little pond, you will see something sports floating like spit on water, some bubbly structure. If you pick them up, you will see that they will be translucent, little eggs, small eggs, anardana ke size, anardana মতন, pomegranate seeds. It won't be there, but it will be whitish. If you keep them in a water inside a jar, you will see that they will little tadpoles will hatch. So there are two hemispheres. The upper hemisphere is known as the animal pole, and the lower hemisphere is the vegetal pole. That is dark. The lower hemisphere, that is the vegetal pole, it is light. So after fertilization it is deposited water and ready for fertilization. You know that fertilization in amphibians is external. The haploid phase, like even our human egg, it is frozen at metaphase of meiosis 2. So once when the sperm enters, a sequence of events takes place. First is that the meiosis, which was frozen at metaphase, this gets completed. Then the cytoplasm of the egg rotates by about 30 degrees relative to the pole. So if you take this as the animal pole, which is dark, and lighter vegetal pole, the top part is known as the anterior, posterior. This is the dorsal, and this is the ventral. Four sides are formed. So once when the sperm is there, this cytoplasm moves 30 degrees. It moves 30 degree move kora jay. Result is a region known as the gray crescent is formed. When the cytoplasm shifts, then a little region known as the gray crescent is formed. In some of the amphibians, Xenopus is like a frog. Uh, it is the light colored band. This is the light colored band which you are seeing over here. অনলাইনে যারা আছে দেখতে পাচ্ছ এটা লাইট কালার্ড ব্যান্ড?
Yes, sir.
Yes, sir.
Okay, a light colored band is formed, and this is formed opposite the point where the sperm is entered. So the sperm has entered somewhere here. Sperm has entered in this region, and this gray crescent has formed over here after moving inside a ball, like a ball sphere. This once this gray crescent is formed, it will tell you where the dorsal is, where the ventral is, where the anterior is, where the posterior is, left kondike, right kondike. So basically, a directional information comes into the zygote. You can see the animal and the vegetal pole, but if you see a ball, just take a cricket ball, on the upper side you paint white, below you paint red. You can say that which is the anterior and which is the posterior, top and bottom you can say, but you cannot say which is the dorsal, the pit, and which is the ventral, the samnes portion. You cannot say that. So the haploid sperm and egg nuclei, they fuse. Already the nuclei has been injected inside, but they have not fused like in fertilization. And it forms the diploid zygote nucleus. Next, you have the cleavage taking place. So the zygote is actually a single cell. From there, you'll have essentially it will become a two cell stage. 2 will become 4. 4 will become 16. Cell will keep on dividing. So by cleavage, that is the cell will divide. Now, as you know, one fact is that the vegetal pole has lots of yolk. Okay. And the animal pole doesn't have lots of yolk. So when the cell divides, the cleavage which takes place, cleavage at vegetal pole is slow. Even before when the first cleavage is done, this cell stage is completed. Already a 4 cell stage cleavage starts to take place. Once the cleavage takes place, you will have small animal pole cells. They are known as micromeres. And large vegetal pole cells containing yolk, they are known as macromeres. Okay. So the furrow, this on the zygote, this furrow starts to take place and cleavage starts to take place. Then 2 cell will become 4 cell. So this is the 2 cell stage. This is the 4 cell stage. Now, third cleavage will take place now. This is at right angles. One took place like this. One took place like this. A third will take place like this. Horizontal. At third cleavage, it will take place at right angles. So result is you have 4 cells on the top, 4 animal pole cells. They are known as 4 micromeres. And we have 4 macromeres below. And this is the 8 cell stage. Micromeres are small in size in the animal pole. Small animal pole, larger vegetal pole. And they have yolk. Yolk laden. Yolk se bhara. Yolk bhara. Then the next few cleavages start to take place, and you have a 16 cell, then 32 cell stage embryo. Then, as the cleavage continues, the animal pole cells begin dividing more rapidly than the vegetal pole and become smaller and numerous. See the animal pole cells over here, smaller and more numerous than the vegetal pole cells here. So after 16 cells, then 32 cells. And you know why the vegetal pole cells divide slowly? Because lots of yolk is there. The cleavage starts takes place late. But animal pole doesn't have, it keeps on dividing. It forms a hollow ball. And this ball is known as blastula. If you take a section, there is a space, and that is known as blastocele. Okay. Then the gastrulation stage will start to take place in frog. Now, here the one thing about gastrulation, sometimes in blastulation, a very important event is some morphogenetic movements take place. Genetic movements start to take place. Now we will go on what are morphogenetic movements. Can you see it? Online wale?
Yes, sir.
Yes,
Okay.
sir.
Yes, sir.
One. So the
Yes, sir.
first morphogenetic movement, say you have a blastula like this. The first morphogenetic movement will take place, that is like a finger will be put inside. The cells will go inside, and this is known as invagination. Right? So you see this movement where the cells, you see that it was zero. The cells they go start to go inside like this. They invaginate. That the word used is invagination. Next is next type of movement is involution. The interning or inward movement of an expanding outer layer. তাহলে interning এই যে the cell has gone inside, এটা ভেতরে এসে গেছে, ঠিক আছে? এবারে these cells, some cells from here gradually will start to go like this. ভেতর থেকে from the inside it will cover the coating. They will divide from the inside. It has already gone inside. This has gone inside. But from the inside now it will start to coat. Another layer of cell will coat. This movement is known as involution. Next movement which we will talk is ingression. That is the cells are on the bottom. They will start moving just on the top like it is shown in this figure here. This type migration of individual cells inside the embryo, that is ingression. Three movements you have seen. Then another important thing, that is the epiboly. বাইরের দিকে. Cells on the covers the outside. Expansion of one cell sheet over other cells. So you have say one sheet of cells over here. Okay. So the on top of this there will be another sheet of cell will come from this direction, or it can come from this direction, whichever direction it will cover from the up. Epiboly is the term we can use, covering. Epiboly, emboly. So epiboly is outside as a unit, and then all the others below, invagination, involution, ingression, they are known as emboly. Here the sheet of cells migrate inside a cell, migrate ইনসাইড আ সেল, মানে এটা ভেতরে হচ্ছে আর ওটা বাইরে হচ্ছে। And so different ways: invagination, involution, ingression. So these are the morphogenetic movements which take place. I have taken this from Gilbert. All these are from Gilbert. This is from a different book, of course, but this is the one which you search. So morphogenetic movements start to take place. I'm just giving an overview of the development which takes place. First, you have the formation of a zygote, gray crescent. Then, after the gray crescent, the division cleavage starts to take place. Micromeres, macromeres are formed. First two cleavages are longitudinal. Next one is horizontal. And you have the over small micromeres formed. Macromeres with yolk is formed below. And then the cells keep on dividing. And after it starts dividing, the animal pole cells divide much faster, so they are smaller and more numerous on the top, and the vegetal poles are cells are much larger and fewer. In after this, 32 around from 32 cell stage onward, it is known as blastula. The blastula has got a fluid filled space in between, known as blastocele. Now, once the blastula is formed, the next thing which is formed is gastrula. So the gastrula formation, we will study about what happens during the gastrulation in frog. So if you see this picture, you just it's a ball. You have taken a section. You can see the blastocele. Blastocele, okay. So these colors are just for actually it is not there in the cell. These colors are not there, just for your understanding. These colors, you can see yolky vegetal cells, the macromeres, the larger and fewer in number. Then you have a region of cell is formed here. These are known as marginal cells. And you have the blastocele in between. Animal pole, vegetal pole. The first thing which will form, that the cells will start invaginating from here. ভেতরে আঙুলটা ঢুকে গেল, invagination. And this hole which is formed, basically I am drawing it like this. So this is invagination has taken place. This hole is known as blastopore. And the lip is known as the dorsal lip of the blastopore. Okay. So let us see what happens. So when the gray crescent is formed, gray crescent is sided on the gray crescent side, that becomes the dorsal, you have seen. So gastrulation is initiated on the future dorsal side of the embryo, just below the equator. Equator হচ্ছে মাঝখানে. So if you see this picture, this is the dorsal side D. So the gastrulation is initiating somewhere here. This is the point where gastrulation initiates here on blastula. This is the zygote which you are seeing. We will cover fate map next day, but today we will see the overview of development, gastrulation, blastulation. So the cells invaginate. আঙুলটা ঢুকে গেল ভেতরে. So if you look at this direction, the cell will invaginate here. And it will form a invaginate and form slit-like blastopore. This whole is, but I'm drawing it like this. This happens in sea urchin, but in amphibia it is more like this. A slit-like blastophore is formed. Then the cell's shape changes dramatically. So in this region, the cells which are present in this region, they become some like a strange bottle. Bottleneck cells. These are known as bottleneck cells. The lining, it becomes the bottleneck cells. If you see the picture, if you see the cells in this region, can you see the shape in that region of the cells? অনলাইনে যারা আছে দেখতে পাচ্ছ shape of the cells?
Yes, sir. Yes, sir. দেখতে পাচ্ছি, sir.
Okay. It's like a
Yes,
bottle.
sir.
These cells are known as bottleneck cells. It's in the covering. They are known as bottleneck cells. And these bottleneck cells line the archenteron as it forms. Then what is the archenteron? This is the primitive gut. Primitive gut means pet, stomach, alimentary canal. So you can already imagine that the primitive gut is going to be formed from here. In the next picture, you can see how the primitive gut has gone. This has invaginated and it has gone from here. This is involution. ভেতর দিকে sheets are sliding inside. দেখো এই cells গুলো they have slide and they have come over here. Structure the cells. These cells from here, which was from here, they have involuted and they have come from here, come to this region. দেখতে পারলে? In the marginal zone, the zone surrounding the equator of the blastula, and when I showed that red colored region is the marginal zone. It starts from there, gastrulation. So the next phase of gastrulation involves the involution of the marginal zone cells, while the animal cells undergo epiboly and converge at the blastopore. So you can see these are the cells which were outside. These are the animal cells, this blue color. These are the animal pole cells. They do epiboly and come over here. Therefore, the blue color has come over here. So the cells have covered it up. And these yellow colored cells, these gray things covering, they have undergone emboli and have come inside. The blastocele has moved. So these are the movements in the morphogenetic movements which are taking place during gastrulation. পরে প্রবলেম হলে ভিডিওটা শুনো, ঠিক আছে? ওই জন্যই I've taken this, I made a brief note for this purpose. Gilbert is a very good book, but Gilbert is more complex in this. The figures are more complex. This is another book which I have taken and pattern, and it is much more simpler, easier. In the examination, this will be easier to draw. Don't have to draw so many little circles and things. You can just shade and show actually the thing which they want to know in gastrulation, whether you know where which cell moves. If you know epiboly, emboli, ingression, where which type of movement takes place, morphogenetic movement takes place, that is most important. It is not important that you show this. It is important that you know about bottleneck cells. You know where the primitive archenteron is. This archenteron, basically, this will form the gut or the stomach and intestine and all this region. So the first cells to compose the dorsal blastopore lip. So this is the dorsal lip. Now later on you will understand that this dorsal lip of the blastopore এই যে ঠোঁটটা তৈরি, একটা ঠোঁটের মতন তৈরি হলো জায়গাটা। This dorsal lip is actually the blastopore. It is known as the Spearman organizer. This is an inducer organizer. It has got certain things. It will induce the nearing cells to form a particular organ. The cells will form the gut. Some cells, if you cut this region and put it in another embryo, it will form the region which is supposed to form over here. This will organize or induce the formation of particular organ, ectoderm, endoderm. So there are organizers. These are the concept of organizers. So this dorsal lip of the so the first are the bottle cells that invaginated to form the leading edge of the archenteron. So these will later form the pharyngeal cells of the foregut. Pharynx থাকে, এই আমাদেরও pharynx থাকে embryonic, gills, pharynx, with this gone. But they'll form the pharyngeal cells, the external, the gills of the frog or tadpole, pharyngeal. Later, and this the dorsal pore of blastopore, the dorsal lip of blastopore is the first inducer. Once this reaction takes place, other inducers on the opposite side, they get activated, they start forming other things. But that is later, just to know what the steps are. So as these first cells pass into the interior of the embryo, the dorsal blastopore lip becomes composed of cells that involute matlab bhetore chole jacche, it's going inside into the embryo to become the precaudal plate, the precursor of the head mesoderm. So it involutes inside. Ultimately, it will form the head mesoderm. Precaudal plate is formed. And all starts from the dorsal lip of the blastopore. That is the most. It's also known as the Spearman organizer. The next cells involuting into the embryo through the dorsal lip are called the cauda mesoderm cells. So first, the precaudal cells, they go inside. They form the. Then the next set of cells which will come inside, involute, are the cauda mesoderm cells. These cells will form the notochord. So পেটের ওপরে notochord will form. Here, in this picture, you can see in the marginal zone, involution is taking place. Lip of blastopore is formed, and this is going inside, involuting inside. That's why green color is showing. The direction on the outside, there is epiboly taking place. Here, there, involution or a type of emboli taking place. ভেতরে ঢুকে যায়. First cells which will involute, they'll form the precaudal plate. Next is the cauda mesoderm cells, which will form the head mesoderm. The widening of the blastopore crescent develops lateral lips, and finally a ventral lips over the additional uh, endodermal precursor cells pass. এই যে গ্র্যাজুয়ালি দেখবে যে এইখানে একটা লিপ আরেকটা লিপ তৈরি হবে, এখান থেকে তো হয়েছে, another lip will form over here and there will be a plug known as yolk plug which is formed. So this is the late gastrula stage. At this point, once it goes inside, at this point, you will have ectoderm opore, mesoderm majkhane, and তলার এই যে cells গুলো দেখছো, they will become endodermal cells, not by the basis of color. Okay. So this is how actually this in frog gastrulation takes place. Chief, let us not disturb ourselves. I will go with this picture once more in detail. From once this is memorized, you can go to the next other minor details. Especially the widening blastopore crescent develops lateral lips, sidal lips তৈরি করে, এটা ভেতর দিকে যাচ্ছে, lateral মানে side lips are formed, বড় হচ্ছে, lips are formed. And finally a ventral lip, মানে উল্টো দিকে, dorsal দিক দিয়ে শুরু হচ্ছে, উল্টো দিকে ventral. So it goes to this side. A ventral lip is formed. এটা দেয়নি ছবিটাতে. Okay. With the formation of the ventral lip, the blastopore এই ছবিটা নেই actually, the blastopore has formed a ring around a large endodermal cells. Blastopore এরকম করে একটা ring has formed around it. So blastopore has gone inside, blastocele has gone inside archenteron, and ওইদিকে first a chorda precaudal cells, then chorda mesodermal cells, then it will wide. This blastopore will widen, and ulto dike on the ventral side there will be another lip which is formed. We will see these pictures later. So far clear, right? So now we will look at what a fate map is. So now the gastrulation process has taken. Now every region has got certain special type of cells. Now what a fate map will say which region will form what. So this basically has been before I go to anything, this basically has been studied by doing certain live techniques. Some cell has been injected with dye, then you follow where the cell goes and what does that form. That dye, by that dye you can detect. This is known as fate mapping techniques. So if you look at the first egg, can you look at this, the blastula or gastrula or whatever happened. So this is the fate map of blastula. This region is going to form the epidermis. This will form the nerve plate or neural plate. This is the suprablastodermal endoderm. This is the blastopore, jeta dekhle, dorsal lip of the blastopore. And this will form the blastopore endoderm. Different types of endoderm. তার মানে this is the epiderm, ectoderm বা epidermis, এটা হচ্ছে mesoderm মাঝখানে, and তলায় হচ্ছে endoderm. Different names. Neural plate is ectodermal. Inside you will see that this region will form the somites and notochord, the mesodermal part of notochord. Little somites are formed during development, little blocks of cells, square blocks of cells, they are the somites. And these mesodermal cells from this region, they will form the heart, anterior blood islands. And this will form the lateral mesoderm, lateral plate mesoderms. And you have the endoderm, it will line the guts. So this is a fate map, what happens. Okay, I stop over here. This becomes very complex now. I will talk about fate maps in details I will talk about this gastrulation process also in details. Some genes and some things are responsible, and talk about the organizers. Then we can talk about that of chick. It's a bit different. In chick, the difference is there is lot of yolk. On top of that, there is only one cell, animal cell, the fertilized zygote. So instead of the whole yolk divide, the yolk stays like this. It will form a blastodisc. The cell will divide. It will divide as a disc. Here it is cutting. Here the cut is taking place only in the animal pole. There's so much yolk that it can't go below. It forms a layer, flat layer of cells. And there all the development takes place. This is the difference between amphibian and chick. Therefore, there you have both of them in the syllabus. Okay, I'm stopping the sharing. Those who are online, could you understand?
Yes, sir.
Questions, Sache?
No, sir.
नो सर আমার নেই স্যার
হ্যাঁ
আমার কোশ্চেন নেই স্যার ঠিক আছে
কি বললো
স্যার আমার কোশ্চেন নেই স্যার আমার
question nahi okay no questions okay fine if you have questions you can ask me later we will go maybe in 2 days 2 or 3 days আমি বলছি either on Monday or Sunday আমি বলে দিচ্ছি হ্যাঁ একটা পরের টাইমেতে বিকেলের দিকে করে দেব হ্যাঁ অনলাইন
ওকে স্যার
থাকবে
Okay, sir.
okay so I am stopping the recording
Thank you, sir.
okay
থ্যাংক ইউ স্যার